The process of translation takes the information stored in the mRNA produced by transcription and translates it into a specific sequence of amino acids. Translation is a three-step process. The first step, initiation, requires three basic components: the mRNA, an initiation tRNA with the amino acid methionine attached, and a small ribosomal subunit. Initiation of translation begins when the tRNA carrying the amino acid methionine binds to a small ribosomal subunit, forming the initiation complex. Next, the mRNA molecule binds to the initiation complex. Notice that the codon on the mRNA that binds to the tRNA is always AUG, which is complementary to the anticodon on the tRNA. The initiation point of translation is always an AUG codon, although this codon can also be found in the middle of the mRNA message. The large subunit then binds to the small subunit. The large subunit has three important sites on it: two tRNA binding sites and a catalytic site. When the large subunit first joins the complex, the tRNA with methionine attached is bound to the first binding site. The second binding site is empty. When the initiation step of translation ends, the ribosome is complete. When initiation is complete, the first tRNA is bound at the first binding site on the large ribosomal subunit, and the second binding site is empty. Notice, however, that the second codon on the mRNA, GUU, is aligned with the empty second binding site. In the elongation step of translation, a second tRNA molecule attaches at the second binding site on the large ribosomal subunit. The anticodon of this tRNA molecule must be complementary to the second codon of the mRNA molecule. When the two binding sites are occupied with tRNAs carrying amino acids, the catalytic site will catalyze the formation of a peptide bond between the two amino acids. The two amino acids are now attached to the tRNA in the second binding site. The empty tRNA at the first binding site leaves the ribosome to be recharged with a new amino acid by enzymes found in the cytoplasm. The ribosome now moves to the next codon on the mRNA, shifting the tRNA attached to the two amino acids to the first binding site and leaving the second binding site once again empty. A new tRNA complementary to the third codon now attaches at the second binding site, and a new peptide bond is formed, transferring the growing chain of amino acids to the third tRNA. Once again, the ribosome shifts to the next codon to be read. This process continues as long as a stop codon is not present at the second binding site. The amino acid chain, a new protein, continues to lengthen. In the termination step, a stop codon enters the second binding site. When this occurs, protein release factors signal the release of the new protein. The ribosomal subunits then separate. As a result, the mRNA strand can be translated again by a different ribosome, and the ribosomal subunits can begin translation on another mRNA strand.